When GDP is bound to the G protein, as shown above, the G protein is inactive. The receptor and G protein work together with another protein, usually an enzyme. When the appropriate signaling molecule binds to the extracellular side of the receptor is activated and changes shape. Its cytoplasmic side then binds to an inactive G protein, causing a GTP to displace the GDP. This activates the G protein. The activated G protein dissociates from the receptor, diffuses along the membrane, and then binds to an enzyme, altering the enzyme's shape and activity. When the enzyme is activated, it can trigger the next step in a pathway leading to a cellular response. The G protein hydrolyzes its bound GTP to GDP. Now inactive again, the G protein leaves the enzyme, which returns to its original state. The G protein is now available for reuse. Before the signaling molecule binds, the receptors exist as individual polypeptides. Notice that each has an extracellular ligand binding site, an alpha helix spanning the membrane, and an intracellular tail containing multiple tyrosines. Binding of a signaling molecule, such as a growth factor, causes two receptor polypeptides to associate closely with each other, forming a dimer. Dimerization activates the tyrosine kinase region of each polypeptide. Each tyrosine kinase adds a phosphate from an ATP molecule to tyrosine on the tail of the other polypeptide. Now that the receptor protein is fully activated, it is recognized by specific relay proteins inside the cell. Each such protein binds to a specific phosphorylated tyrosine, undergoing a resulting structural change that activates the bound protein. Each activated protein triggers a transduction pathway, leading to a cellular response.